recap where we are getting back into chapter 11, which is acknowledging the efferent subdivision of the peripheral nervous system. And we're going to contrast our somatic nerves from our autonomic nerves. Last time that we met, we were talking about predominantly the autonomic nervous system, and then today we'll round out with the somatic nervous system. Keeping in mind the efferent nervous system. So if we're looking at the spinal cord, which side of the spinal cord would we be looking at? The ventral side. Okay, so we're looking at all nerves ventral if we're looking at the spinal cord, efferent nerves. Whether they be voluntary somatic or involuntary autonomic. We're looking at the efferent control. Is descending, ascending implied in this image? So are descending, or?
Yes.
Kind of. I would see the brain and the spinal cord, but we don't see any direct relationship in this image. All right, so y'all okay with where we're going to study in chapter 11? Efferent nervous system. First involuntary, and follow up with the voluntary nervous system. We introduced this term called dual innervation on the last time we met, and I'm going to kind of flesh it out a little bit more, but I asked you to read ahead in chapter 11 so we can talk a little bit better today about it. What did we learn about dual innervation? It means that these nerves generally oppose one another. What else?
Most internal organs are dually innervated.
Very good. Most of our internal organs are dually innervated. It's almost all of them, but there's a select few that are not. Okay, so most of our internal organs are dually innervated. What else am I going to add about it? What does that mean when she said most organs are dually innervated? What does that even mean? Some organ, some effector organ, muscular gland, is dually innervated by both subdivisions of the. What do you mean by both? Let's clarify that.
By the
Autonomic.
Very good. Both subdivisions of the autonomic nervous system, which is the sympathetic and parasympathetic nervous system. So a lot of our effector organs, heart, liver, stomach, intestines, keep going, are dually innervated by both subdivisions of the autonomic nervous system, which we're going to call sympathetic and parasympathetic. So it tells us that we can control these organs whether our body is in the phase of what? Fight or flight, or even relaxing, right? I can control my heart if I'm taking a nap, or my body can control my heart if I'm taking a nap. And if I want to run, nothing's going on. No. If I want to run, which nervous system can control my heart? Sympathetic, right? So we can dually control organs under periods of rest as well as under excitation. But it takes specific nerves. Because each nerve releases a different neurotransmitter, which gives a different message at the target. The effects of one another tend to oppose one another. So sympathetic nervous system does what to the heart? Excites it, speeds it up. Parasympathetic must slow it down. We could just keep going on with these opposing effects. So before I move on, what questions do you have about the difference between dual innervation or even these opposing effects of the sympathetic versus the parasympathetic? All right, and then we left off looking at the anatomy. And in discussion in class, in my section yesterday, they're having real trouble making the connection with Chapter 5. So let's just make a connection to Chapter 5. Chapter 5, what were we studying in Chapter 5? What were we studying in Chapter 5? Chemical messengers. Okay, we said neurotransmitters, hormones, neurohormones, periphenotically, right? Well, we're just taking that one neurotransmitter and expanding upon it, right? So we're looking at extracellular messengers and neurotransmitters, but we're studying, well, how do they get released? What are they? What's their effect? Okay? And so some terminology we used were source and target. We also looked at the solubility of a messenger. Is it more hydrophilic? Is it more lipophilic? Right? That indicates how long it gets released, where the receptor is, what the receptor probably does. Right? We covered all that. Now we're just looking at specific examples. So don't just completely try to separate chapter 5 from chapter 7 and 8, wherever we're in chapter 11 here. It's just unfolding from what we introduced. And so I tried to illustrate early in the semester, this class just builds, it layers. And so we can't just, that's chapter 9, done, right? Whatever. We're just building all semester. And so instead of saying source and target, because that's super generic, well, now I can say pre-ganglion cell. I can say post-ganglion cell. Instead of saying neurotransmitter, I can say acetylcholine. Instead of saying receptor, I could say cholinergic receptor. And that tells me what the signal transduction is. If you remember in chapter 5, I said the cell does something. And here we're saying, well, acetylcholine binds to a nicotinic receptor. The target cell will depolarize. And we're just going to say it does something. Does that make a little bit of sense? The connection? Hopefully the connection are kind of made here. And if we use this terminology, in the autonomic nervous system, we learned that the anatomy is unique, and what we thought before, in that in order for our peripheral organs, our cranial, our spinal nerve, to control our effector organs, it's not one nerve that extends to the effector, but it's actually how many? Two separate nerves that extend, then we approach the effector organ. We gave some specific terminology focusing on this area. What did we label this first area as? A ganglion, okay? Which we said is a collection of cell bodies. So if we can locate the ganglion of an efferent nerve, we know there's two nerves in a row. So the first one's called the preganglion cell. And the other one is called the postganglion cell.
Now you said there's two on the...
1, 2. And then you get to the effector organ. Okay. Heart, smooth muscle, whatever. So, parents, where were you going?
No, I thought I heard you say there were two out there on the post.
No.
So the two
no.
make up one.
The two cells would collectively be some autonomic efferent nerve. Whether it's whether it's sympathetic or autonomic, whatever it is, there's going to be a preganglion and a postganglion cell, which then itself will innervate the effector organ. Additional clarification?
Did you say that in the autonomic nervous system it has a short preganglion and a long post?
That is dependent upon if you're looking at the sympathetic or parasympathetic.
Oh, okay.
The length of the nerve depends if you're looking at the sympathetic or parasympathetic nerve. So without quantitating it, either way, if there's two in a row and you see a ganglion, you're looking at an autonomic nerve.
Mm-hmm.
But involuntary control or some part of that. Or not. Additional clarification? What neurotransmitter will the preganglion cell always release? Acetylcholine. Very good. The preganglion cell always releases acetylcholine. I don't know if there's an exception. I don't think there is any variation in there that I know of. And so it's always going to release acetylcholine. So without getting specific, just generically looking, what type of receptor should be there? That'd be the specific one. So generically, we call them
Nicotinic?
Well, the nicotinic would be the most specific. If I just said generically, I would say a cholinergic receptor. I was going to ask you first cholinergic, but you guys know the specific time to say nicotinic. So you can say nicotinic cholinergic receptor, which binds to acetylcholine and does what? When acetylcholine, a chemical messenger, is there, what do the nicotinic receptors do? They cause an excitatory response in the postganglion cell because how are these things built? What is the nicotinic receptor? Is it binds acetylcholine? And my question is, why is it excitatory? What makes it?
It's a sodium potassium channel.
It's a sodium potassium channel. It lets more sodium into the cell. Into the postganglion cell, which causes the excitatory. So the nicotinic cholinergic receptor is just an example of a chemically gated ion. When acetylcholine is present at the postganglion cell, it will depolarize to its threshold and generate an action potential. If acetylcholine is released, the postganglion cell will generate an action potential. We need to ensure that the message is conveyed all the way to the effector. We don't want any decision making going on in that area. Keep the message going. The postganglion cell gives us some variation though. It may release what?
Acetylcholine.
It may release acetylcholine. It may even release epinephrine, or may even release norepinephrine. Will it release any of the three?
Either one.
One of them. A nerve will release either acetylcholine or norepinephrine or epinephrine consistently. It's not going to just pick and choose today what it wants to release. So, like the vagus nerve, whatever, right? Just picking out nerves, they will release a known neurotransmitter. But not all of our nerves release the exact same. And so if we got some variation of neurotransmitters released by the postganglion cell, what else do we give variation to?
Receptor.
The receptor is variable then, as the target tissue. If the preganglion cell releases acetylcholine, the postganglion cell needs a what kind of receptor? Cholinergic, whichever one, I don't know. If the preganglion cell releases epinephrine and norepinephrine, if the postganglion cell releases epinephrine or norepinephrine, the effector cell would be which type of adrenergic? Adrenergic. Good job. Good job. It needs some type of adrenergic receptor. Alpha adrenergic, 1, 2, beta adrenergic. Those all activate second messenger systems, so then they can go in the back of it. It'll target cell and do something. What questions do you have about the relationship of an autonomic nerve, about the term preganglion, postganglion, the neurotransmitter release rate, and the receptors along the way? Okay? Come back to that. If we look at figure 11.1, this will help visualize the dual innervation. On the far left, you have the sympathetic nervous system highlighted, and on the far right, you have the parasympathetic nervous system highlighted. The sympathetic nerves are colored in green, where the parasympathetic nerves are in the purple color. And if you have your book, you go to see it obviously a bit better. But if you take any given organ, say the heart, it is what? Dually innervated. So is the excuse me. So is the liver. You go through. So is the pancreas, right? We can begin to see some exceptions. So are the intestines and the large intestines, etc. There are a few exceptions.
Why would your bladder need to be dually innervated?
Well,
I'm sure the reason is obvious. I'm not getting there.
So think about it when you're resting and digesting versus if you're doing something like fighting and flighting. Fighting and flighting, You want that bladder active? No. One is a rest, right? But when you're resting and digesting, we need to excite those muscles. So we need to have the dual control. Relax, contract, or evacuate. So we want to be able to control those parts. Even let's go here, right? Even more dramatic. Let's dually innervate that. So now I'm fighting and lose all control. Do I?
Be a defense mechanism.
Actually, that is a published defense mechanism for, I didn't realize this, but the military teaches for assault, sexual assault, so just pee all over them. So if you can. Hopefully it works.
I could totally backfire on you.
Not making light of this scenario, but peeing on somebody else. It's an interesting new defense tactic.
I think like animals actually do that.
Let's turn back to dueling. Alright, so a lot of our internal organs are dually innervating. The question is like, well, why? You can normally be about resting. We need to control it as well, suppress or whatever the case may be when you're fighting the additional questions? There's always exceptions in this class, right? There is a structural exception to the postganglion cell in this thing called the adrenal gland. The adrenal gland is an organ which is on top of the kidneys, and we're going to see it now, and we're going to see it when we come back from spring break when we talk about hormones. And so this is a cool structure. It releases hormones and neurohormones into the circulatory system. And this is where we got hung up yesterday. So let's not forget, we're looking at extracellular messengers. And so if something goes in the blood, it has to first do what? Be reabsorbed. And where we got hung up is, does it need a carrier? Would an amino acid base something need a carrier? No. Okay, so, and is the blood vessel the target for? No. That was just, y'all got it. So the blood vessel is not the target for where we're going. It's just a transport mechanism. Okay, so let's elaborate this. We're going to look at the adrenal medulla, the middle of the adrenal gland today, and then we'll come back from spring break. We'll look at the adrenal medulla. And if you look at the adrenal medulla, you have a sympathetic nerve. The preganglion is shown here in blue. So the preganglion, presynaptic cell releases what? Acetylcholine. The postganglion cell has what type of receptors? What type? The nicotinic cholinergic receptors. But if you look closer, I don't see a postganglion cell, right? So it's considered to be modified. The structure's a bit different. So the postganglion cell is this kind of blue-colored structure in this image. They're called chromaffin cells, and the chromaffin cell is it acts as a postganglion cell. But if you look at it, it doesn't look like a nerve cell, right? It's completely different. Okay. But it still functions like a nerve, so it releases what?
Norepinephrine.
Neurotransmitter, you guys, epinephrine, norepinephrine into the circulatory system. So this is how it's sympathetic. So when you are, how do we categorize this? When you are excited, fighting, whatever, your adrenal medulla releases epinephrine into the blood, and it just goes system-wide. So when someone talks about their adrenaline rush, they're talking about the epinephrine spewing, I guess, from your adrenal medulla. When you fight or flight, epinephrine goes in the circulatory system. And then, of course, we learned that it would be filtered out to all your target tissues, the brain, the skeletal muscle, the liver, right? Just keep it on. You can actually feel it, right? If you ever had that adrenaline rush, you just feel the physical changes that are going on in your body. It was induced by adrenal medulla epinephrine. we're getting system-wide instead of just a tiny little synapse here. And so therefore, because it goes into the circulatory system, we can call this a neurohormone. I think the doctors can't call it neurotransmitter. You can call it. We had that debate earlier. Is it a hormone? Is it a neurotransmitter? What is it? So let's make up a new word and call it a neurohormone. It's a blend between the neurotransmitter and the from the nerve, but it goes into the blood. We'll call it a neurohormone. This is in my distribution. So the acknowledgement is that the adrenal medulla acts as a modified post-ganglion cell. It releases excitatory neurotransmitter into the circulatory system. You okay with that? Any questions before we move on? Some other modifications which you may see about the autonomic nervous system, summarized in figure 11.9. Up till now, we said a cell has an axon terminal where neurotransmitter is released. And if you look at the anatomy of some of our autonomic nerves, they release neurotransmitters not just at the end, but along the length of part of the axon. And those allow for the release of neurotransmitter, not just in one spot, but in a bit of a wider spot. But the target is still adjacent. So it's not going into the blood necessarily. And those areas are called varicosity. Varicosity is just an autonomic nerve that releases neurotransmitter in a wide area. And maybe use the word like diffuse spot, or diffuse and spread out, right? Instead of just a tiny little, one little spot, you may have it all over. So one nerve can control a lot of cells simultaneously, autonomically. Y'all want to take that too? So talk about varicosity. What is that?
Relation of transmitter. In a lot of places.
In a wide area. Very good. Question we got yesterday, does this have anything to do with blood vessels? And the answer would be just no. Simply, varicosity, I don't know what the word translates to, but it gave kind of, this is a nerve, not a what? Not a vein, right? And so I don't know what the word translates to, but I'm going to guess it has something to do with this widening or something. So a blood vessel could also bulge, right? But this is intentional. Okay? So these varicosities are intentionally there to release neurotransmitter like in a large area to control the receptors. Any questions over that? All right. Taking it easy on me. And then as well, we did not talk about the central nervous system outside the lab. And so I'm going to call your attention that this doesn't just happen, it is controlled by the central nervous system, integration center. And a lot of our autonomic control comes from the adrenal, not the adrenal, but the medulla oblongata in the brainstem. And so I have specialized cells in the brainstem that give that dual innervation control over many of our internal organs. So, for example, we'll come back to the brainstem, for example, in the respiratory system. And we have parts of the respiratory system that excite and allow for respiration to go up when you're running. And we also have the inhibition center that slow down respiration. Same thing with the cardiovascular system. We have areas that excite the nerve that control the heart, and we also have areas that inhibit the nerves that control the heart. So what we're doing is just acknowledging those autonomic nerves, get their control, and they're from the central nervous system. The etheric nerves, their job is just to keep the message going. And the message originates here in the central nervous system. So we'll use this respiratory center, cardiovascular system, center terms at the very end of the semester. Come back to the right. In table 11.3, to read the whole table in your textbook. I just highlighted a few of some examples. We've already had some examples in our discussion of dual innervation, looking at whichever effector organ, contrasting the parasympathetic versus sympathetic nervous system effect. So we're talking about the heart, is how the heart rate can decrease under parasympathetic control, yet increase under sympathetic control. I don't think we necessarily addressed the muscle part of the heart, but the muscle heart is also innervated in that when the parasympathetic nervous system is in control, the heart contractility is a bit weaker. It's not weak, I mean it still works, right? But it's not as strong as when we see the sympathetic nervous system, which makes sense. If you've ever run, right, you can feel your heart like, oh, boom, pounding, right? So it's being fast and more forceful, under control of the sympathetic nervous system. Your pupils, the muscles that control your pupil diameter, they will constrict when you're resting and digesting, so you're not taking in much food, so like relax, sleepy, right? And then contrasting that to the sympathetic nervous system. Your pupils will dilate, you're alert, you're taking in as much information as you can in the chat to the body. And then I don't know that this is, I don't recall the liver necessarily being in that list, but I think it's an important thing to acknowledge, is that the liver is also innervated, and under the parasympathetic nervous system control, our nutrients are retained. We're conserving, you're probably maybe eating, so we're taking nutrients out of blood and storing wherever we can, including the liver, adipose tissue, or whatever. And so the parasympathetic nervous system actually conserves nutrients. What does the sympathetic nervous system do? Releases them, and even maybe promotes their catabolism. So get that sugar out of the liver, and you'll start powering your skeletal muscles. You get heart muscles, brain. So it releases glucose. And if you recall, we talked about glycogenolysis, gluconeogenesis, those types of things might happen to release glucose out of your circulatory system so you can power our body. Those are just some examples of the opposing effects of the sympathetic and parasympathetic nervous systems. And if we go back addressing time, I guess, as far as how these regulate the body, you remember we said the nervous system is fast, right? But if we contrast the parasympathetic and the sympathetic nervous systems, they both will release their neurotransmitters quickly. However, how our body responds to them, how long, I guess we should say, our body responds to each is variable. And what we'll find is that the sympathetic nervous system effects are more long-lasting than the parasympathetic nervous system. And the reason why, it goes back to, first, epinephrine goes into the blood. So it's not just a microscopic synapse, it's system-wide. So it's just going to be in the blood as long as until it's used or broken down. So it's going to remain present for quite a bit longer than if it were released into a synapse. You also should recall that our catecholamines activate a second messenger system, which we know takes a while, but also the effect is amplified. So, all that business, right? It amplifies a little bit of time. So it's a lot. A second messenger takes longer than just opening and closing an ion channel. So it prolongs the effects. And then because the messenger goes into the circulatory system, it's just going to take longer to break down, to reach the liver, to reach metabolize, to process. So this might make more sense to you to give you an example, right? Right now, which part of your nervous system should be predominated? The parasympathetic. Rest and digest, pay attention, take notes, do an exercise, right? And so we're maintaining it, no problem. What if we're sitting in here, we hear a door slam, boom, and someone says, fire, run! What's going to happen? Sympathetic nervous system is going to kick in fast, right? Yeah. Don't carry you out if it's not. Alright, so it should kick in, right? The sympathetic nervous system should, in a moment's notice, quickly take over that rest and digest and alert. Heart rate up, respiratory rate up, glucose into the blood, right? And let's say that happened. Let's say it really happened. And then so we calmly exit the building, And then we get out there and someone's like, April Fools! And, right, so then you come back to your room in here. Can you as quickly chill as you got excited? No. It's going to take you minutes. And even if you start thinking about it more, like how we're going to dot that guy's eye, right? For doing that April Fool joke, you're going to continue, right, to release that epinephrine. But even once you try to focus, it's still going to take a few minutes. To remain alerted, yeah, you never know when another scenario may happen. But the point of that analogy is that the sympathetic nervous system can quickly override the parasympathetic nervous system. Yet, the parasympathetic nervous system cannot quickly alter the effects of the sympathetic. It would probably be pretty dangerous.
It's like whenever you're in PE and the teacher tells you don't just immediately stop running because it's not good for your shins or something. Like, just, yeah.
And so same kind of thing, right? It would be dangerous on the heart to increase the cardiac, what we'll learn called cardiac output, the blood that is pumping out, and then just, you have all this overload of blood and deficiency coming in, it would just be a big mess, I think. So I agree, it's a good thing. Good. Additional comments or questions? That looks like that concludes our introduction into the autonomic nervous system, and we'll transition now to the somatic nervous system, which is which nervous system? Our voluntary nervous system. It gives us voluntary control over which types of organs?
Skeletal muscles.
Skeletal muscles. You're going to like this one a lot better compared to the autonomic nervous system. It's a bit more simple in contrast. But either way, it's a form of efferent control, right? So if we're looking at the spinal cord, which side of the spinal cord are we on? Still on the ventral side. But we're looking at different nerves with the effective organ. So it's still controlled by the central nervous system, interneurons, etc. If we look at the somatic nervous system, how many nerves extend to the effector? One. Alright, one nerve, and it always releases acetylcholine. So no variation there. Our somatic nerves release acetylcholine. Our target is the skeletal muscle would be the target because it has the receptors. What kind of receptors should it have? The cholinergic, definitely, and also the nicotinic receptors. Your skeletal muscles that are under voluntary control express nicotinic receptors. Which we know has what type of effect of the skeletal muscle? Excitatory. It's always excitatory. The somatic nerve excites a muscle, the muscle should contract. And which we'll pick up in chapter 12. A little bit easier, right? Somatic nerve always releases acetylcholine. Skeletal muscles always have nicotinic receptors. And so when acetylcholine is present, a skeletal muscle will, even before we get to contraction, the skeletal muscle will do what? It's excitatory. It's going to let which ion in? Sodium. So the skeletal muscle will, what's the word? Good job. Depolarize. Good job. Skeletal muscle is going to depolarize. So that tells us we're going to keep on using potential charges terms after this class. If we look at the arrangement of our somatic nerves, they're not quite as simplistic as maybe implied in this image. A single nerve may branch out and control multiple muscle cells at the same time. And so our axonal terminal branches to different cells. So maybe not just the same muscle cell, maybe control maybe 5, 10, 20, 100 muscle cells simultaneously. And the range was considered to be a motor unit. The motor unit is a term to describe one somatic nerve and however many muscle cells it's working with. So it's how many nerves? 1, and then X number of muscle cells. So a motor unit is one somatic nerve and then however many muscle cells it's branching out and controlling. So how many motor units are shown here? There are 3 shown here. Motor unit 1 looks like it's controlling at least how many cells? 4. At least 4, yeah, in this image. Motor unit 2 looks like it's controlling 3 for its skeletal muscle. Well, we'll extend this when we get into the muscle physiology. Like, why do we care about this? We do care about it. It's going to affect how much force that muscle can generate. We'll continue that in chapter 12. In about a month, we'll visit it. I just want you to be familiar with the word motor unit. Really with the somatic nervous system. What questions do you have about that terminology, motor unit? I'm going to introduce another new term to you, maybe. And I don't want you to get hung up on the familiar word. The word that's going to be familiar is innervation. So let's just address what the word innervation means. We used it for dual innervation. What does the word innervation mean? I'm going to not go in as
It has a nerve connected to it.
Innervation.
It has a nerve connected
It has
to
a
it.
nerve in it. If something's innervated, it has a nerve. Okay, so it's just a generic way to say there's a nerve in there, whether it's afferent or efferent. Whatever. And so if I use the word dual innervation, though, that gives me these things have nerves and we know what kind though. Are autonomic nerves. We can use another type of innervation to illustrate the somatic nervous system. And we may see some reciprocal innervation of our somatic our skeletal muscles. Some of our skeletal muscle organs are reciprocally innervated. What does the word reciprocal mean? Opposite, right? Some skeletal muscle organs are reciprocally innervated by the somatic nervous system. What that means is some motor units are recruited and some motor units are turned on, the others are turned off, suppressed. But they happen at the same time. Well, that doesn't make sense a little bit, but maybe if it makes sense if we understand a bit more about our body, right? Your muscles that you study in anatomy are muscle organs. And so if I say muscle organs are reciprocally innervated, I activate one muscle organ and inhibit another. Which makes sense. If I want to move, I do something simple. I've got muscles on groups on both sides of my bones. So when I do a movement, I need to excite some while relaxing the others. And so if I want to move, I've got to have some reciprocal innervation. If they're both exciting at the same time, am I moving? If they're both relaxing at the same time, am I moving? No. And so I need to have some reciprocal innervation. Does that make some sense? So reciprocal innervation is just the term to describe how our somatic nervous system excites some nerve or some muscle cells while hitting others so they can do movement. Whether it be voluntary or even like we'll see in the middle of a reflex. Which is illustrated in this image. Before I move on to show the image, what question do you have over this term reciprocal innervation?
It's kind of like that whole like antagonist muscle thing.
Yes, that'd be how your antagonist muscles would work. and they'd be reciprocally innervated. Very good. And I've got an old image, but I think it works. So, for example, if you put your hand, like you do a quick reflex. Textbooks love a hot stove, right? Don't you know, nervous system work. So some stimulus, an afferent nerve, activated by interneurons, which activates specific efferent motor nerves. If you want to get your hand out of the way, you don't want to contract all the muscles, right? You want to relax some and contract others. And so if I want to pull my hand out of the way, which muscle group should be excited? The bicep should be excited while the tricep group is inhibited. So we can turn them off. So we are y'all okay? All right. We have already kind of introduced what happens to the skeletal muscle when it receives a colon. We said it's going to depolarize, it's going to get excited because our nicotinic receptors are basically what? They're chemically gated sodium channels. They're not calcium. Calcium involved is not yet. A whole other segment. But the first thing is acetylcholine binds to the nicotinic receptors of the skeletal muscles, and sodium floods into the skeletal muscle. And so skeletal muscle is going to work. All of our muscles are going to start out like a nerve. They're going to depolarize, cross threshold, generate an action potential. So the story will be redundant when we look at each of our three types of muscles, and that's what happens here in the skeletal muscles as well. So first we grade up to what? Threshold. Then we open up our voltage-gated sodium channel so we can generate an action potential. And I've got a bunch of stuff because I think it's about a week control. Okay, so eventually we'll leave the contraction as well. So just to remind you, after we take our test on Tuesday, we're going to pull in that extracellular messenger of our hormones. So we're going to do endocrine system, then back to muscles. So don't lose the ground you're making in this first unit over potential change. And so your textbook goes through and kind of puts a visual on this. What type of nerve is this? A motor neuron, a somatic motor neuron, very good. And the heat shade would be the skeletal muscle. That was kind of on the control. We know that our somatic nerves release acetylcholine. We know that our motor nerves have nicotinic receptors, which causes polarization. Please don't hurt them. It's already great job. I think that concludes our content for Chapter 11, which I wanted to present to you. So I want to open it up for questions over Chapter 11, autonomic versus somatic nervous system. Before we go, we do have plenty of time, I think, to talk about the replay. Don't worry about labeling. Okay. Yeah, don't worry about labeling all the events. The big thing is we have acetylcholine binding to nicotinic receptors, which cause the cell to depolarize. The rest of the stuff you will cover, trust me. This is figure 11.15 in your textbook.